29th. Dr. Mark Williams, he's been here several times and gives great talks. They're typically on some kind of current medical concept, something that's hot in the news, and he's going to talk about Ebola. So I would encourage you to come. Anybody have any questions? Anything that you need to say out loud? Nope.
Be on time.
Be on time.
Early is on time, on time is late, late is late.
Okay, we're in the process of talking about cell organelles. And we ended the last time talking about the mitochondria, didn't get quite through the mitochondria. Remember, your kind of word association is mitochondria, energy production, specifically ATP. We're going to see later on that the process that produces ATP in the mitochondria is a pretty interesting phenomenon called chemiosmosis. And in chemiosmosis, we're going to have for this to work, two spaces separated by a membrane. The mitochondria has a double membrane structure. If we use this room as the analogy for the mitochondria, the walls being the outer membrane, outer row of chairs being the inner membrane, matrix in the center, that's our first space. And then this space between the outer membrane and the inner membrane, called the inner membrane space, is the second space. The reason that we need these is that during chemiosmosis, the mitochondria is going to develop a hydrogen ion gradient. Anytime we're talking about a gradient, we're talking about something at a low level and something at a high level. So we're going to develop a high level of hydrogen ion concentrations on one side of that inner membrane and a low concentration at the other side of the membrane. That represents potential energy. And we need potential energy to make ATP because the process of phosphorylating or adding a phosphate group onto ADP to make ATP is energy requiring. So there has to be an input of energy, and that input of energy is provided by that hydrogen ion gradient. So one of the things we're going to spend some time talking about is how in the mitochondria is this hydrogen ion gradient established? What's responsible for forming that? And then how is that tapped as an energy source to make the ATP that's necessary to sustain all living organisms? So we're going to come back and talk about the mitochondria in a lot more detail later on. This is an interesting, somewhat colorful picture taken by some advanced type of microscopy using some fluorescent dyes. And one of the things that this really illustrates is that the cytoplasm of the cell is filled with this meshwork of protein molecules that constitutes an internal cytoskeleton. The importance and the significance of the cytoskeleton is a relatively recent understanding. I think I mentioned before that when Dr. Morton and I were taking these classes, we really didn't know anything about the cytoskeleton. We didn't really have the tools to be able to visualize that very well. One of the things that is really important about that cytoskeleton is that it's highly dynamic, in that these tubules of proteins can assemble into long rod-like structures and then later can disassemble and take itself apart. And so there's this phenomena going on constantly within the cell where the cytoskeleton is constantly changing and reorganizing itself. And you saw some of that in the images of the inner life of the cell that I showed you. And one of the striking images I thought was that assembly and disassembly of those cytoskeletal elements. There are three types of elements that make up the cytoskeleton, and they are the microtubules, the microfilaments, and the intermediate filaments. We're going to take each one of these and look at them kind of individually and dig into them a little bit. As a whole, the cytoskeleton has a lot of diverse roles that it plays in the cell. For example, one of the things that it does is it helps to maintain cell shape. It's responsible for cell motility. In those cells that are contractile, like muscle cells, the cytoskeleton is the basis for that particular contraction. And it's also responsible for organelle and vesicle movement. And that's one of the things I want to talk about just a little bit right now. We talked about the endomembrane system, and we said that the endomembrane system was the series of membranes within the cell that were either physically connected to one another, like the ER membrane and the nuclear membrane, or were connected via those vesicles, those little transport sacs that could pinch off of one membrane system, like the ER, and then fuse with another one, like the Golgi, or the plasma membrane. Okay? But those particular vesicles typically aren't just kind of floating around randomly. Instead, there are some very important mechanisms within the cell that allow organelles to move in a kind of a purposeful or directional way from one set of membranes to the other. One of my favorite images in inner life of the cell is this big sac with what it looks like two little feet at the bottom of it. And that's an organelle with some motor proteins attached to it, and they're crawling along or moving along those cytoskeletal tubules from one destination to another. And so this business of organelle and vesicle movement really has to be coordinated for the cell to work properly, and the cytoskeleton really helps to make that possible. Let's start out with microtubules. They are the largest element of the cytoskeleton. Microtubules are proteins that are folded up into a cylinder-like structure so that basically it is hollow in the middle. It's almost like that little paper tube that's inside of a roll of paper towels. Okay? The outside of that tube would be the amino acids strung together in the polypeptide chains, making up those proteins, and then they're folded into that helical or cylindrical shape with a tubule in the middle. And those are the microtubules. Here is a portion of the cytoplasm of the cell showing you a portion of the cytoskeletal system. And you can see these large diameter microtubules make up this internal meshwork that permeates through much of the cytoplasm of the cell. Now, there's some details here that you don't really have to memorize for an exam. They are hollow tubules. They're made up of a particular type of protein called tubulin. Don't memorize diameters. Okay, you'll see some other reference to diameters for other cytoskeletal elements. You should know that these are the largest element in the cytoskeleton. Okay, the microtubules have the biggest diameter. Okay, what are their main functions? Maintenance of cell shape. It's kind of like an internal support of girders in the cytoplasm of the cell that helps to maintain the cell in a particular shape. And remember, not all cells have the same shape. One of the things that someone commented on when I showed some images of cells the first day was how those neurons had these long projections, the axons and dendrites of their plasma membrane, and they extended well away from the body of the cell. That is all internally reinforced by cytoskeletal elements. We're going to talk about the role that the cytoskeleton plays in cell motility, specifically cilia and flagella. I mentioned organelle movement. One of the elements of the cytoskeleton that you are somewhat familiar with from last semester is the role that the cytoskeleton plays in chromosome movement during cell division. Okay? Remember in prophase that we start to form that spindle apparatus, and that the chromosomes attach by their centromeres to those spindle fibers, and by the spindle fibers moving and contracting and telescoping inside one another, line up the chromosomes on the equator of the cell, and then start to pull them apart. Okay? Well, prior to that happening, the cell's internal cytoskeletal system completely reorganizes. And it disassembles and then reassembles into these structures that facilitate chromosomal movement. If the cytoskeleton is responsible for maintaining cell shape, then sometime during the process of cell division, you would expect a dramatic change in the shape of the cell, wouldn't you? Because this structure, this system that maintains cell shape, is being reorganized into another purpose. And that is something visibly you can see in cells as they prepare to go through mitosis, is the cells undergo a pretty significant shape change. This shows you a stained, fluorescently stained look at cells, and you can see the filamentous nature of the cytoskeleton in green right here, blue's nucleus. Here are those cytoskeletal elements, the microtubules, and then this gives you an idea of how individual protein molecules of tubulin can all be joined together to form this hollow cylindrical tube that forms one of the major structures of the cytoskeleton. Cytoskeletal elements are also involved in centrioles. Centrioles are structures that are found in animals but not in plants that help to organize the spindle apparatus. So that's something you might have heard of before from last semester. I want to spend a little bit of time talking about the role that the cytoskeleton, and particularly the microtubules, play in cell movement involving both cilia and flagella. And this is pretty timely because this week in lab you're looking at some cells that are modal. For example, your paramecium moves very rapidly by beating of those cilia. What's typically the distinction between a cilia and a flagella? What's the difference between the two? Cilia tend to be shorter and more numerous. Flagella tend to be longer and less numerous. Sometimes a cell will only have a single flagella. Cilia and flagella both work in the same fashion. These particular structures move by undulating back and forth, and that creates momentum that then propels the cell throughout the environment. One of the most studied flagellated human cells is the human sperm. And movement of this particular single flagella is very efficient in moving that sperm to its ultimate destination, fertilizing an egg. Do we have any ciliated cells in the human body? Males have flagellated cells. Do we have any ciliated cells in the human body? I see some heads nodding. Yes, where? We do. Digestive tract has peristaltic motion, which is muscular contraction, which moves things throughout the intestine. Go a little farther up. Yeah, in your trachea, even down into the bronchi, there are epithelial cells with numerous cilia, and they beat back and forth. What does that do for you? It creates really a wave of motion that kind of sweeps particles up and back up toward your mouth. So if you inhale or ingest things, that there is a mechanism to sweep them back out again. It's sometimes referred to as the ciliary escalator. And it kind of is like moving things up through an escalator. That sweeping motion keeps things from settling more deeply down into the tissues of your lungs. If you're a cigarette smoker, which I hope none of you are, chemicals in cigarettes, including nicotine and tars, can paralyze cilia. And that's one of the reasons why cigarette smokers have a much higher incidence of lung infection than non-smokers do, is it compromises that particular defense mechanism of your body. Here is a single-celled organism filled with multiple cilia on its cell surface. And again, the motion of this cilia is moving backwards and forwards, side to side, propelling the organism. As it turns out, prokaryotic cells can be flagellated as well. But if you take microbiology, one of the things you'll find is that even though these two structures are both involved in motion, prokaryotic flagella and eukaryotic flagella, they're organized in very different fashions, and they have different mechanisms of movement. The eukaryotic cilia and flagella move back and forth, side to side like this. The prokaryotic flagella is kind of like a prop on a motorboat engine, and it rotates around like this. But we're going to just talk about eukaryotic cilia and flagella now. Okay, we'll skip by there. If you look at a cell that has cilia in this particular case, what you'll see is that those cilia are really just little projections of the cell membrane. It's kind of like a little pocket that's been pushed out of the cell membrane. But then inside of that are these girders, if you like, elements of the cytoskeleton that fill the inside of that little membrane-bound sac. And so this is a pretty rigid structure. So what makes up this cilia is cell membrane on the outside, and then these proteins of microtubules on the inside. The microtubules are arranged in a pattern that is referred to as a 9+2 arrangement. If you take a cross-section of the cilia, you can see what that 9+2 arrangement looks like. Okay, so again, here's our cell. Here is a single cilia. So here's our cell membrane. And then here's microtubules inside of that piece of membrane. If we then do a cross-section through that, this is what it looks like. There are 9 doublets of microtubules around the outer edges here. And then there are 2 single microtubules in the center. So that is the 9+2 designation. And then you can't really see it here, but then there are some little cross-linking proteins that join the inner 2 tubules to the outer ones. And then in red here, another protein that's really important. That protein is called dynein. Sometimes they're referred to as dynein arms. So in order for this particular structure, the cilia, to impart momentum, what it's going to have to do is it's going to have to bend back and forth from side to side. Okay? And that will impart momentum. This is work, and so it's going to require ATP. So cell movement consumes ATP. That's one of the reasons we have to have cell respiration, so cells can move. Mechanically, though, how is this accomplished? How can some ATP and some protein tubules result in a flexion of this structure back and forth, side to side? Well, one of the keys to making this all work is the phenomena of phosphorylation and dephosphorylation. Let's say I've got a protein molecule, and I add a phosphate group to it. Chemically, a phosphate group becomes attached to that particular protein. Adding a phosphate group is phosphorylation. Where does that phosphate group come from? It comes from ATP being broken down into ADP and phosphate. So the source of the phosphate is ATP breakdown. Okay? When I phosphorylate that protein, it changes its shape. Now, this is going to be a really important concept in a lot of things we're going to talk about this semester. That change in shape is called a conformational change. And conformational changes are what drive a whole bunch of types of cell work that occur within cells. So if I phosphorylate this protein, it shifts into this conformation. If I dephosphorylate the protein, if I take this off, it shifts back to this shape. So a molecule like a protein can alternate between two forms. This form and this form. This form and this form. Depending upon whether it has been phosphorylated or dephosphorylated. Phosphorylated or dephosphorylated. Okay? This is going to be something that's going to be really important when we talk about transport across the cell membrane. And some of the transporters in the cell membrane that pump substances across the membrane do so because the transporters become alternately phosphorylated and dephosphorylated. Well, what's happening here in the cilia is that these particular molecules that make up the cilia, the microtubules and the dynein arms, interact with one another differently depending upon whether these proteins, the dynein arms, are phosphorylated or dephosphorylated. If they become phosphorylated, these dynein arms change shape. And literally they move and kind of grab the microtubule at a different place than they were attached before. And that bends it. And then when they become dephosphorylated, they move back to their original position. So it is this alternate phosphorylation and dephosphorylation of these proteins, the dynein arms, interacting with these tubules that ultimately result in that flagella or cilia flexing back and forth, side to side, side to side. So, yes, ATP provides the quote-unquote energy for this particular phenomenon of movement to occur. But ultimately what the ATP is doing is it is providing the phosphate groups, which ultimately are the mechanism by which these changes in shape are accomplished. Okay? So it's not really kind of the same as burning gas to make a car go. Instead, what this energy molecule is doing is breaking down and producing the phosphate groups, and the phosphate groups attaching, releasing, are ultimately what is responsible for doing the work. So that's an important distinction. As I said, organelles are not in fixed position within a cell. They do move around. Here's an example of a vesicle. And here is a microtubule that could be part of the cytoskeletal system. The vesicles will have little receptors on their surface for proteins called motor proteins. And these motor proteins are a lot like the dynein arms in the cilia and flagella. When they're phosphorylated, they change shape. When they're dephosphorylated, they go back. And so by alternately phosphorylating and dephosphorylating this motor protein, you can get this walking motion where the vesicle is moving along that cytoskeletal element towards some particular destination. It's almost like a track, a rail track in the cell. It provides a guide for those vesicles to move from one location to another. So here's an actual electron micrograph. Here is the cytoplasm of the cell, and here's a microtubule. And you can see these vesicles that are in the process of walking along this particular cytoskeletal element towards some particular destination. Second type of cytoskeletal element are the next smaller elements, the intermediate filaments. These are fibrous proteins that are supercoiled into what look like thick cables. So each of these little protein subunits is wrapped around another protein molecule. These aren't hollow like the microtubules are. So they're smaller in diameter, they're not hollow. They also help to maintain cell shape. And they play an important role in anchoring the nucleus, particularly to particular locations within that nuclear membrane. And this is something we won't get a chance to talk about, but the organization of the nucleus and how those chromosomes are organized within that nuclear membrane is a factor that really influences how genes are expressed, whether they're turned on or off. And so these particular proteins actually do play a role in gene expression regulation within the cell. Here's something that you'll look at in lab this week. This is a intestinal tissue. Here is our lumen, or the cavity of the intestine. And here are some of the epithelial cells with those little foldings of their membrane, the microvilli. Here you can see the cytoskeleton and how cytoskeletal elements help to maintain the kind of shape of those particular little projections of the plasma membrane, those microvilli. And here are the intermediate filaments. There are either some smaller filaments involved in this called microfilaments. And those are the third element of the cytoskeleton. These are the smallest in diameter. Here's an example of how a microfilament might be organized. A couple of polypeptide chains wrapped around one another. So you don't have this cable-like arrangement where there are multiple chains of polypeptide. Typically there's only a couple of them wrapped around one another. The primary component of microfilaments is a protein called actin. Have you heard of actin before? Where have you heard of actin? Actin and its partner, myosin, are really important in muscular contraction. They also play a role in cell shape. They play a role in a phenomenon called cytoplasmic streaming. They play a role in cell motility, including amoeboid movement. When you see your amoeba move this week, it's going to be because of changes that are occurring in the actin portions of the cytoskeleton. And microfilaments also play a role in the formation of the cleavage furrow during cell division. Remember in cell division that once your chromosomes have segregated, that the cytoplasm kind of pinches into two, and you literally form this furrow in the cell that separates it into two compartments, the cleavage furrow. That's the result of actin filaments interacting with one another. The interaction between actin filaments and myosin filaments is crucial in muscle contraction. As these slide past one another, The muscle cells can shorten and lengthen, giving us muscle contraction and relaxation. What's probably going to be involved in this movement of these filaments past one another? Myosin and actin phosphorylation and dephosphorylation. Okay? So whether these are phosphorylated or dephosphorylated will affect how these proteins interact with one another, and they'll slide past one another, and that gives us our muscle contraction. So the physiology of muscle cells is incredibly adapted to that particular structure or that particular function of muscle contraction. This is showing you amoeboid motion, what you'll see with some of your amoeba, and how little extensions of the cell can kind of flow out. They call these pseudopodia. That means a false foot. And this is the result of reorganization of the actin portions of the cell cytoskeleton. In plant cells, we're not looking at any, we can't get L and D anymore, can we? One of the things that you oftentimes see in plant cells is that the cytoplasm moves in a circular fashion, and you can tell it's moving because the chloroplasts are moving within the cytoplasm. So you can see this cytoplasmic streaming within individual plant cells. That's also the result of actin filaments interacting with one another in particular ways. Let's get over that right now. Okay, so everything we've been talking about so far are structures that you would find in a quote-unquote typical animal cell. If we're talking about plant cells now, which of these apply? Which of these organelles and structures, all of the same organelles and structures that are in plant cells, that are found in animal cells, are found in plant cells, plus which one of these are also found in in plant cells? Sorry. Mistake on the slide. Should be one more choice. What should the choice be? All of them. Okay. In addition to all of the organelles we've talked about so far, plant cells will typically have a cell wall, a large central vacuole, and chloroplasts. Okay? This is something that is a common misconception that we need to correct. The misconception is that animal cells have mitochondria, plant cells have chloroplasts, but no mitochondria. That is incorrect. All of the eukaryotic cells have mitochondria, whether you're plant or animal. But additionally, plant cells have chloroplasts. Okay? So here's our kind of composite plant cell. You can see this very rigid box-like structure that is found external to the cell membrane, and that is the cell wall. What's the cell wall made of? It's made primarily of cellulose, and what cellulose? What kind of a molecule is it? It is a carbohydrate. It is a polymer of what unit? Glucose, right? How are those glucose units bonded together? By alpha bonds or beta bonds? Beta. Okay. What's starch? It is a carbohydrate. What is its subunits? Glucose. How are they bonded together? Alpha. Okay. What's glycogen? Sometimes called animal starch. That is also alpha glucose polymers. Does anybody know the name of the enzyme that breaks down the glucose polymers in starch and glycogen? It's called amylase. Okay, and to break the alpha bonds, you need alpha amylase. Do we have alpha amylase?
Yeah.
Yep, we can digest starch and glycogen. What enzyme do you need to break down cellulose? Beta amylase. Do we have beta amylase? Therefore, we can't digest cellulose. Are there organisms that have beta amylase? What does? Okay? I hear cows. Do cows actually have beta amylase? Nope. So how do cows digest cellulose? They have microorganisms living in their mini-chambered stomach, and those bacteria have the enzyme beta amylase. So the bacteria digest the cellulose, and then the cows live off of those digested materials. Okay? So the cows really are eating the bacteria. What about termites? Do termites have beta amylase? They don't either. In their guts, they have protozoans, and the protozoans are what produce the beta amylase. What's the purpose of the cell wall? It will maintain shape, yes. It helps to determine cell shape. It also does something else really important. And that is, it helps to protect the cell from swelling up and bursting. Why might a cell swell up and burst? Well, we'll get there in a second, but plant cells are typically filled with this large central storage area or vacuole, and inside of that vacuole is water. And it is not atypical for a plant cell to have water rushing into its central vacuole, and the vacuole get bigger and bigger and bigger. And without the cell wall, what would happen? It would burst. It would lyse. Like the paramecium if the contractile vacuole wasn't working properly. What's the cell wall do? Well, it's this rigid box outside the cell. And as the cell starts to swell because water enters the vacuole, what happens? The membrane pushes out against the cell wall, and the cell wall exerts inward pressure, and eventually an equilibrium is reached where the vacuole can't swell any longer, but the cell wall exerts enough inward pressure to prevent the cell from bursting. So the cell is nice and swollen or turgid. What's the advantage of cells being turgid? What happens to plant tissue when this central vacuole isn't full of water? It shrinks, it shrivels up a little bit. And then what happens? There's no outward pressure exerted on the membrane, so the membrane starts to pull away from the cell wall. And the plant tissue wilts. Okay? Because that cell is filled with water and is swollen, but the cell wall protects it from bursting, plant tissue is nice and turgid, and that gives it structural integrity. So a leaf, for example, can be outright like this, as opposed to all curled up and wilted up like this. When you're turgid, the tissue's turgid, that gives the leaves maximum surface area for absorbing sunlight. So it's almost like an internal support system. It gives a plant tissue integrity. Okay? Kind of like our skeleton does for us. So the cell wall is really important in maintaining cell shape and preventing cells from bursting when they're filled with water. The structure of the cell wall can be fairly complicated. There can be several different layers of cell wall material. There are primary cell walls, there are secondary cell walls. We're not going to worry about that too much. One of the things we'll see later on is that it's important for plant cells to be able to communicate with one another. And if each one is surrounded by this box, that could kind of hinder cell interaction. So there are some adaptations that allow cells to communicate with one another by actually joining their cytoplasm across that cell wall. So we'll come back and talk about some of those connections a little bit later on. Here is an electron micrograph of a cell. So here's our cell wall out here. You can see our chloroplasts. You can see the cytoplasm. And then there is this large central vacuole that is filled with water and typically other dissolved substances as well. So if that vacuole is filled and there is outward pressure exerted on the cell, then this cell is nice and turgid and the tissue will be erect, have structural integrity. We'll talk a little bit later on when we talk about osmosis and diffusion, why water typically tends to move into that vacuole, and therefore why plant cells tend to usually be turgid. In addition to water, there are a number of solutes that are present within that vacuole. Some of those solutes might be pigments. And next week, are we doing the beet experiment? Okay, next week you're going to be working with beets. And we're going to be doing some investigation of cell membranes and vacuole membranes. We're going to be looking at a pigment in beet cells called beta-cyanide. That is located within this vacuole. There could be various metabolic wastes that accumulate within those vacuoles. And some of these metabolic wastes are poisonous compounds. If they're segregated within the vacuole, then the cell cytoplasm is protected from those particular dangerous substances. Can you think of any advantage of a plant cell accumulating these poisonous, toxic substances? There's a whole class of compounds called alkaloids within the vacuole. Okay. Yep, protection. Okay. Plants are not mobile and they can't get up and run away from predators. But by accumulating toxic substances in their vacuole, if something eats them, it could kill them or make them sick. And if it makes them sick, hopefully the predatory animal will learn to avoid that particular plant. So it's an important defense mechanism. On the average, plant cells tend to be a little bit larger than animal cells. Remember what we said limited cells from getting too big? What puts a limit on cell size?
Surface area.
Surface area to volume ratio, right? And when we're talking about volume, we're talking about cytoplasm, aren't we? Well, one of the reasons that plant cells can be a little bit bigger than animal cells on the average is because this central vacuole takes up so much space that there's really less volume of cytoplasm. So even if the membrane is a little bit bigger, the volume hasn't increased proportionately because of the presence of this particular vacuole. it's not uncommon to see a central vacuole squish the cytoplasm of a cell into this little kind of thin layer all around the edge of the cell. So even the plant cell's bigger, its surface area to volume ratio is still favorable enough because of the effective decrease in the amount of cytoplasm. A plant organelle that we'll spend a lot of time talking about later on after we do cell respiration will be the chloroplast. And obviously, the word association for this particular organelle is photosynthesis. The chloroplast is really a pretty remarkable cell organelle, because ultimately what it's able to do is to absorb sunlight energy and then convert that absorbed energy into chemical energy. The initial products of photosynthesis and the absorption of light involve production of ATP and a compound called NADPH2. This particular compound is a reductant. We'll talk about that later on. These two forms of chemical energy allow carbon dioxide and water to interact to form glucose. So this is a very complex function. The structure of the chloroplast is going to be then adapted to carrying out this particular function. So if you look at the chloroplast, both in artist's rendition and in an electron micrograph, you can see that it has a fairly complicated structure. It has several membranes. It's got actually two outer membranes. It has got an inner membrane system, and those inner membranes are stacked on top of one another, kind of like stacks of coins. Inside of this outer membrane, the thylakoids are floating in a cytoplasmic-like material called the stroma. When we talk about photosynthesis, we're going to see functionally that photosynthesis is broken down into two processes. One is light dependent and the other is light independent. They're going to occur in different parts of the chloroplast. The light dependent portion of photosynthesis is associated with these membranes or thylakoids. The light independent portion where the carbon dioxide and water actually form glucose are going to happen in the stroma. The light-dependent processes produce ATP. And ATP is made in the chloroplast by the process of chemiosmosis. Same process that produces ATP in the mitochondria. In order for chemiosmosis to occur, we need two compartments separated by a membrane. The membranes are these thylakoid membranes. And in the cross section here, you can see how there's a space inside this membrane, the way it's organized. So there's a space, a membrane, and then the stroma out here represents the second space. Why do we need that? Because we need to establish a hydrogen ion gradient to make ATP, just like we do in the mitochondria. So the advantage of when we do respiration is if you get comfortable with chemiosmosis in the mitochondria, it's a piece of cake to understand how that happens in the chloroplast as well. Same basic phenomenon. Okay? Done. We will continue on, probably wrap up with this chapter Thursday. I'll give you your next reading quiz assignment on Thursday. Okay?